Okay, let's take a look at student questions from last week during a break. First question. So, this lady was a little uneasy about this using GFP because, like GFP gene, using GFP, I thought GFP is a unit that can glow itself. Yes, a sign. Yeah, exactly. And then shouldn't GFP be attached to the proteins then composing the cells I want to track, not the genes? Uh, yes, GFP is a protein. So every protein is made from what? DNA, gene. So if we want to attach a protein to an existing any protein, practically speaking, the easiest way and also reliable way, technically speaking, at the current level of technology, is the fusing at the DNA level is the easiest way. So you just put together those two genes and then create a chimeric gene, and then this chimeric gene will be converted into, will generate such chimera protein. That's how you do it. So you start from the DNA, the gene, then it will eventually make the corresponding protein, and that's why you do it. A DNA guy was fun. Yes, I'm a DNA guy. Why is it funny? DNA guy is some the weirdo. Yeah, DNA guy is a weirdo. So this is maybe yeah. I have to admit it's funny and true. Um, and so yeah, last week I mentioned that my ego as a DNA guy something to blah 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 something. Yep. So ego as, but ego kind of those the English speaking people use this ego a lot, like as if the ego is a synonymous to pride, right? So yeah, as if everybody has their own version of pride. DNA guy also has his or her own pride. That's why I. And if we introduce genes that make unique enzymes of archaea into a human genome, can humans survive extreme environment like archaea? I wouldn't say it's not possible. Yeah, it is possible. Somehow. I don't know how, but somehow. But before we can achieve such level, probably if we keep trying to become like archaea, like by incorporating more and more of those archaeal genes to resemble their ability, probably our entire functionality will first become like entirely collapse into those of archaea. That's probably my more immediate worry. So maybe possible, but practically at this point still like unrealistic. Although someday, eventually, we can pinpoint actual the difference and the reason how and why archaea can somehow manage such extreme environment, and then somehow we can better adapt it someday. I believe, as a DNA guy, someday I believe that they will come. Uh, the archaea and bacteria are totally two different species. Yeah, it is not species, but try to remember this. Here is a common ancestor, Luca. So many different big groups. And these areas are eukaryote. Eukaryote. These big groups are archaea. And these big groups are bacteria. So these two big groups, such big groups, we even call them as a domain, not even species. Species are a little bit, such a little tiny, and these are the prokaryotes. And that's why I mean, archaea. There are intrinsically two big difference exist between even they are prokaryote. Both of them are prokaryotes. These two big groups of organisms are different enough that we cannot put these two groups into a single same group. That's why we have to separate this. Okay? Not even at some kind of a species level, but as such a huge group scale, we call domain. Yep, your hard prayer has been answered. So we are going to have a take-home exam again. I like that. So do you have any favorite microscope or experimental equipment? Kind of out of context, but I remember the yeah, your high school chemical teacher favorite was what, a pipette. Hey, this is my lab bench. How many pipettes I own? These are all my personal pipettes. Yeah. So I do have a luxury of different pipettes. Nobody else can touch. These are all mine. It's upstairs for the thin. So I have enough pipettes and many other equipment, and also have really many fancy equipment, including, so for example, like fluorescence microscope. I always think that, oh, I wish I had such a fancy fluorescence microscope right beside this lab bench. It would be really convenient. But there are so many different such equipment that I wish to have, but I don't have. But actually, if I think only one, I have one such imaginable equipment that I really would die for have. Guess what? Robot. I'd like to have my robot so that I always tell the robot that, hey, do this, do that, so that I don't have to worry about actually doing this thing, my actual experiment. I truly, firmly believe that actually that day will come, sooner or later. All those lab work will be actually handed over to these robot things. Yeah. The humans sitting there and sipping coffees and do this like mental work, and robot will do actual physical work. That should be the way of lab work, I believe. How do you distinguish between characters and trait terms in Korean? Yeah, it's a good question, because that's what I have wondered too. Because if you look up the dictionary, both character and trait are in Korean. It's been translated as a 형질 trait. Now, how can you function with this two different categories of definition using one single word? So actually, there's been compromise. Where are you? Oh, shoot. Oh, yeah. I didn't put it. So in some textbook, they try to distinguish this character versus the trait as a character as a the literal translation of character as a this particular thing, whereas a trait as a this 형질 thing. That's in some rare occasion. That's what I have found the way they distinguish these two confusion. Uh, is there any specific role for the intergenic region? Yes. Let's say some partial area about DNA, and let's put this red area as a gene region. So the rest of the region would be intergenic region, but still, so before, like in the past, some 20 something years ago, most of the scientists would consider this intergenic region as a simply garbage area, but no longer. uh, do they think? Uh, these even intergenic regions, a lot of times it's been found that they have a very important critical roles in controlling the actual gene region. And besides, these intergenic regions, a lot of those intergenic regions actually produce the non-coding RNAs of many different kinds. So if that is the case, intergenic regions role has a very significant, some specific roles there. During the lecture, you mentioned that we can adjust the light wavelengths, but on Google, it says the light wavelengths can be controlled. Of course, yes, light wavelengths can be controlled by using simply different filters, light filters. So we can specifically choose one specific portion of light wavelengths to have. But the problem is, to visualize a sample, we need entire mixture of light wavelengths, the panorama of light wavelengths. If we simply choose only one particular wavelength of light, usually we have a difficulty in visualizing. That's what it is for our eyes. So that's the practical problem. Can every different species that have a same chromosome have offspring between them? No, it is not, because just because you have a same chromosome number doesn't always that you are guaranteed that you can have an interbreed. Chromosome number differences is a first barrier. If you have a different chromosome number, then you just forget about it. However, more actually important thing about than the sheer chromosome number is this homologous chromosome. The reason why you have to have the exactly same chromosome number is like one-on-one. One-on-one, you have to have one homologous chromosome from which you can produce the haploid number of chromosomes. And each haploid number of chromosome now will join and to find their respective partner as a homologous chromosome partner. But consider that organism A, organism B has the same chromosome number. What do you mean homologous chromosome? Homologous chromosome means if you have two chromosome aligned, homologous chromosome, at each position, each location of a chromosome, you do have to have exactly same kind of genes, like the same characters, should be distributed. Only then you have any meaningful this pairing things. Just because you have the same chromosome number, like you have a 3 and 3, but each chromosome does not match in terms of the location of the gene, then you cannot have this kind of pairing things. So, chromosome number, having equal chromosome number is obviously, of course, you do have to have one particular prerequisite, but more than that, more importantly, you would have to have exactly same type of homologous chromosome matching. Then there is no barrier, at least. There's no barrier, at least for this type of the reproductive isolation mechanism. There's no barrier. You are confused with the difference between genome and gene. The difference between genome and gene can be compared with like a forest and tree. If the tree is a gene, then the entire forest will be a genome. Easy. So a gene is an individual tree. The entire forest having all different kinds of tree is a genome. Yep. That was a quick. So I have to stop for a moment for this recording and resume because still we have still more than 10 minutes before I start again. Or should I just start early and then finish it early? What do you think? Which one? It's up to you. But do I have to go immediately move on to the second session and then finish early? Or you still have 10 more minutes of relaxation? What do you think? The what? Finish early. Yeah. Any other opinion? Finish early? Sounds good? Okay. Uh, so this epigenetics can be achieved by simply this adjusting this gene expression status like that. And acetylation, methylation, forget it. It's a chemically how you can achieve different status of epigenetics. Uh, but yeah. So, but the thing, important thing is, if you become euchromatin, which means you are more relaxed, uncondensed, then now the genes on the same that area will become activated. What do you mean activated? Like they will make more amount of DNA. The transcription activity will become higher, more active. Yes. So it is tightly linked. Euchromatin, higher gene activation, higher gene activity. And vice versa is true. If you become heterochromatin, that area will become more condensed. Okay. Then gene, we call such status as genes are silenced. Zip, which means you lose, temporarily speaking, you temporarily lose your ability to make DNA. So that's how we describe as their gene activity has been lost, or their gene activity has been down. Okay. And this epigenetic, who controls this? Whether this particular area will become euchromatin or heterochromatin, who affects, who control these things? Is among many things, yeah. Many different factors can control and affect this, whether a certain particular area will become hetero or euchromatin, but one such factor that can control is the environment. That's how we can relate also like this races as a racial difference as a epigenetic kind of a epigenetically controlled feature or trait is because races are what? Races are usually become established in particular area, local area, live there for a long period of time under the specific environment and then eating some specific food and having some particular exposed to very specific particular environment and somehow their appearances and other traits has been fixed there. So it actually fits into this type of the observation characteristically speaking. Okay, and so environment means we usually link this environmental influence as an acquired trait, isn't it? So if some trait has been the result of the environment, then we just simply label that as, oh, that's an acquired trait. Because that's something that is not due to your gene change, like DNA sequence change, but simply as a consequence of the adaptation to specific environment, and you just like downplayed that as just an acquired trait. Like remember this giraffe's neck, acquired trait, you cannot do anything, right? But now think again, because This epigenetics, a lot of this epigenetics, like as a consequence of this environment, which means acquired trait. And now some of them, some of such epigenetic traits are, look at the racist thing. Racial differences are something, obviously something that has been kept and inherited into next generation, which is we can think only as a consequence of some specific environmental factor, then it should have been acquired trait, but those traits has been passed on to the next generation. Likewise, it's been it turned out that many such epigenetic traits can be transmitted onto the next generation, even though those epigenetic traits are actually the consequences to the response to specific environment, but that somehow kept recorded as a memory in our cell, and then specific status of gene expression are also passed on to the next generation. So now what? Literally, acquired trait is being passed on to the next generation. Okay? So Lamarck, yeah, we once we call Lamarck as a liar, or like at least those who like spread the incorrect information. We have to think again, yeah, revisit Lamarck, and then at least for the at first we have to say sorry, apologize, and okay, you're right, at least partially is what. So what's the bottom line is no one knows exactly what's true in science. That's the beauty of especially the life science. It can change in any minute. The more you study, the more you get confused, and you have to be really ready for accepting some new doctrine, new idea. You have to be really open-minded. Anything can be possible because you never know. That's something that you firmly believed no longer true in a minute, depending on. Okay, so now then, how do you actually control the gene expression? Because it's very important. How to control the gene expression, it makes a huge difference in terms of many things. So it's no longer always only the DNA sequence change due to the mutation, but without it, you can still make huge difference by adjusting the gene expression. That's my area, actually, my personal research area. How to adjust some certain gene products, gene expression, so that I can achieve some difference. Anyway, that's my personal. Once again, that's my personal ego. So you have to ignore them. So this is kind of a general statement. Everyone knows by now. So gene expression, as I repeatedly stated today, like it's an activity of making RNA from DNA template. So DNA, RNA transcription, I have already said that. Now the thing is, even there are some genes, but not everybody is like actively engaged in RNA making business. Some genes are active, some genes are inactive or silenced. So these active genes can be labeled like actively making many copies of RNAs. Inactive genes are making virtually no copies of RNAs, like that. And also, it's very weird in that this activity can change over time or over different area in our body, okay, or under different environment. Okay. Yeah, it's not strange, of course, because we already have recognized that this gene expression can be controlled by environment. Environment can change, so this gene expression can change. Okay. timely. Also, this gene expression pattern, when to produce and when to shut off, is actually tightly and wisely programmed. Remember progeria? Okay? Why we suffer such deadly consequences when we lose the integrity of the nuclear envelope membrane? Because we lose such a fine programming. When to express, when to shut off, and totally messed up. So you suffer such a grave consequences like that. So it's been timely, well programmed, this thing. And also, different location have a different gene activity is also you can easily recognize. Okay? Here, some specific gene must be produced to generate some specific type of cells. In our blood, some specific gene must be expressed like hemoglobin. Okay? Even though hemoglobin genes is everywhere, but only place that hemoglobin gene is active is in our red blood cell. Yeah, strange, isn't it? Why do we simply, if the rest of the other area of the cells don't need any hemoglobin gene, why do we still keep them? Simply dump it. You are not going to use that hemoglobin gene ever for the rest of your entire life, but still you keep it, but simply making them silenced. And that's everywhere. So each different part of our cell as a multicellular organism have a different preferences for what kind of genes are going to be expressed, whereas what kind of genes are reduced, like a shut, inhibited to for this expression like that. So this is a concept of a differential gene expression, enabling us multicellular organism. The differential gene expression is the mechanism behind this differentiation. This differentiation is the key requirement for becoming a successful multicellular organism, isn't it? Yeah. You have to follow this rule of differentiation so that every different part of your cell should be different in terms of different tissue, different function. Okay, as a different tissue, you have to play different role and you have to look different. Right. How do you achieve is this? The answer is by employing so-called this differential gene expression, like hemoglobin. That's why only hemoglobin gene is expressed only in the red blood cell, whereas some other genes are expressed only in that particular area. Or time-wise, timely, only certain genes are expressed when you are only baby and then silenced. And some other genes are when you become juvenile, okay? Then only that gene become active. And some other genes are like when you reach the age of some like middle age or you get old, then some genes are become more actively engaged like that. Okay, so who is the guy responsible for this making RNA? So gene activity. Gene activity means by the how much RNA you make is the parameter. Like we can determine like gene activity. So if certain genes, the RNA copies being made, so many copies, then you are so active. And if some other gene area of the gene makes such a low copy of RNA, then you are so poor. You are inactive type of. Who make this difference? Who is the guy? You wanna know? Enzyme, of course. RNA polymerase. Yeah. Although we don't have to know the specific name, but for each different activity, there is a corresponding responsible enzyme. And in this case, RNA polymerase. Right. Fine. So this guy will scan this area and excuse moi and open up and then make copy complementary copy of particular strand of these two strands, pick one strand and then make a copy, RNA copy is basically DNA. Hey, the reason why this guy lose my sensing is I always make so many hand move. So this guy got confused of my this hand. But how can I just, maybe I have to tie up my hands and then, okay? All right. So, but this is actually something very interesting. So gene, hey, everybody wants to become a gene. Yeah, if you are part of a DNA, yeah, you rather become on some kind of an important area than nothing. So probably I can assume, my ego said, I can assume that everybody wants to become a gene. Then how can I become a gene? Not everybody can become a gene, but actually only very small certain specific area function as a gene. Then what's the difference between this area that serves as a gene and the area that cannot serve as a gene? The difference is the presence of this particular sequence element called the promoter. Promoter is like landing spot for RNA polymerase. So if you luckily have this ready for this promoter, like sequence, promoter is the DNA area of DNA, like let's say this is the part of a gene, this is a start, and this is the end, the DNA, so this DNA will make RNA, okay, the corresponding size of RNA, then promoter is usually right up here. This is the promoter area, okay? So this promoter area is not found on the actual product of the gene, which is RNA, even though RNA is the copy of the gene, but it does not include the promoter because promoter is outside of actual this so-called coding area or coding region. But this promoter is extremely important. Without it, this area cannot function as a gene. Why? Because for every gene, it needs the RNA polymerase, and to lure RNA polymerase into this area to work, you need to have this particular landing spot for RNA polymerase, which we call promoter. So now, if you have a promoter, RNA polymerase can sit over here, RP, then now RNA polymerase can start this thing. So that's a simple thing. If you want to generate any piece of DNA to work, to function as a gene, then all you gotta do is, okay, you prepare this DNA sequence, whatever it is, and then place this particular DNA sequence element, which is promoter, place together and then put back into your genome, and it will work as a gene now. Very easy. Okay. But for those areas who don't have this promoter, it's a really, really shame because I want to work as a gene, but simply the only thing that I lack is this promoter. So anyway, so this area promoter, but also for typical eukaryotic gene, you have more than promoter. You have so many additional some DNA element you better have to work efficiently. Because not only this RNA polymerase, but you do also need some friends of the RNA polymerase to work efficiently. Because RNA polymerase, if you imagine the way RNA polymerase work, It is not something that your gene is laid on the floor, like sitting on this floor. So your RNA polymerase job is walk on this solid flat floor and doing the job. No, it is not. If you think about it in three-dimensional way, RNA polymerase work is something like can be compared as an acrobat, like rope-like gene in the 3D way, and this RNA polymerase is like climbing, scanning, and opening up this rope, and then trying to assemble. So it can easily fall out of this rope. Think about it. That's the way it is. So to support such dangerous job, yeah, you need a lot of those friends to have a stabilized thing. So anyway, so you do need some other additional helper proteins for this for efficient gene activity, and those helper proteins are called transcription factors. Simply, those are the proteins that need for efficient transcription for RNA polymerase to work. That's it. So actually, now people have realized that the difference between active gene and inactive gene is not the presence or absence of a promoter. No, because the difference between gene and non-gene is actually presence and absence of a promoter, right? If you want to become a gene, then you better have absolutely the first requirement is you have to have this promoter. But for every gene, yeah, every gene has this promoter. If you have promoter, then RNA polymerase will visit. Fine. But then still, why some gene is active while others are inactive? What's the difference? The stability of this RNA polymerase. In some gene area, they don't have any friends. This RNA polymerase will walk alone, lonely, and then it falls very soon, and then so on. For some other genes, there are many supporting steps. Transcription factors are there, okay, and cheering for and stabilizing, so RNA polymerase can stably move along. So it's transcription factors. This is the one that actually make the difference between active and inactive gene. It's not the RNA polymerase per se. RNA polymerase is everywhere. It's a one core element which every general gene will have. But the difference is whether any particular gene can become active or not is determined by whether you have a transcription factor or not available. And that's the actual area where why euchromatin has such a high gene activity, whereas a heterochromatin has a low gene activity. Because euchromatin, they have a more access because it's an open world. So welcome, my friends. So this transcription factors can easily come and have access. This tight, squeezed, condensed area, just the rocky area, there's no landing spot can be found for these different transcription factors. So this transcription factor, even if they want to help, but they cannot land. So, yeah. So actually, that's the physical difference. Before they didn't realize, but nowadays they realize that, oh, in order to simply just control this different gene amount of gene expression, you actually whether this area is a heterochromatin or euchromatin is the foremost important factors. Of course, yeah, other factors can also be involved, but the most important thing is this structure, right, between hetero and euchromatin structure. Why? Because of this transcription factors accessibility. Okay, can be determined by this physical. Yeah, it is some difficult concept, isn't it? However, if you somehow can follow this concept, and all of a sudden you actually your the knowledge level about at least about this gene expression, understanding this gene expression can certainly become a way up high. This certainly, believe me. So at least that's probably something that you can have as a take-home message or a reward for today's lecture. So I will probably you realize that I'm just like cutting out unnecessarily detailed, complex things out of these things to make it simpler. So just bottom line is we talked about transcription factors and their importance. So if you had a transcription factor as a protein, then your gene expression will become higher. Okay, so become active gene. If you don't have them, you become silenced, inactive gene. That's it. Other miscellaneous details things you try to find personally, or you can ask me individually later if you are further interested in. Now, another thing about this gene is let's consider this area. So, right below the promoter, after the promoter, this is the 1 unit of the gene. I'm talking about the eukaryotic gene. So this area will correspond to the actual RNA being made. Okay. And if this RNA is going to be used in protein making, then this RNA is what? What kind of RNA is this? At least that has to, you have to know. If this RNA somehow happens to be the one that is going to make the protein, then what kind of RNA is this? mRNA, yeah, exactly. There is only one kind of RNA, which is coding RNA. So this must be an mRNA, right? So this belongs to the case of mRNA. Now, in eukaryote, what happens is, so this is a primary. It's called the primary RNA transcript. Transcript is something that transcription product, transcript. So it is equal, it is synonymous to RNA. You examine this RNA, primary RNA, and the final version of this RNA is this. Mature, finalized version. So this RNA, messenger RNA, this mRNA goes through some editing procedure until it finally become a mature, final version of this RNA, this. Between the primary, baby, and adult RNA, what's the difference? Can you tell the difference? Obviously, size is different. Why? Why this adult RNA become shortened? By simply looking at this cartoon, can you tell what happened? Yeah, in terms of what? Yeah, something labeled as introns are all gone here. Introns are all disappeared. And what else do you have in the primary RNA? In addition to introns, what else do you see in primary RNA in labeling? Exon, yeah, exon. So this primary RNA seems to be something that is like made up of exons followed by introns and exons followed by introns and exons. That's it. And somehow, by going through this, whatever the editing, all introns are gone, eliminated, Then what? The remaining exons are all put together to make a final version. That's how you do this, the final version, make the final version of eukaryotic messenger RNA. Prokaryote don't have this kind of complex business. They have a more straightforward way of making RNA, but this is the case of a eukaryote messenger RNA. So introns are gone, exons are put together. You insult me? So coding region consists of two different area segments called exons plus introns. So to define an exon, probably by stating segment of an mRNA coding region that remains in mature mRNA is probably one of the way to define exon. Then probably the way you can define intron might be segment of an mRNA coding region that is no longer that exists or that is removed from mature messenger RNA. Fine. But the thing is, why? Why do we have to go through this silly business? The introns are the things that we don't need, right? Then why are they in the first place, making this thing unnecessarily complex? It's really mind-boggling questions. Okay. Probably, if we have time, enough time, probably we can revisit and then talk about this. At least, even though we do not know exactly why, still we keep this introns, but still by keeping this intron, what we can achieve is probably what we can at least talk about a little. But before that, first let's take care of this thing first. So this differential gene expression, which is the hallmark, characteristic hallmark of all this multicellular organism. But if you ever wondered that how this so-called differential gene expression is accomplished, okay? So this is the mechanism, very simplified version, of course, version of what we have discovered, what they have discovered, not me, what they have discovered. And so behind this thing, what else? The transcription factors were the one who were responsible for this differential gene expression. So we now, assuming that we know that, at least understand that transcription factors can make the difference between gene expression, high and low gene expression. And by employing this, differential gene expression means, like, for example, let's say one such example of differential gene expression would be different gene expression in different location of our body. Like some like tissue specific gene expression. Tissue specific gene expression. Certain different organ or tissue will make preferentialism specific gene expressed while some others are silenced, whereas some other area of your tissue will have the opposite or something comparable thing is we can define as a tissue specific gene expression. And if that pattern is showing over the timing, like when you're young versus when you become adult, then we call that as a developmental specific gene expression. But the same thing, yeah? Whether you control this locally or whether you control this over time, this thing. So let's take a look at the example is a local difference. Like here, two gene. One, the gene name is albumin. It's very famous protein, albumin. In our blood, albumin is one of the major protein present in our blood. Okay. But actually, this albumin gene is made by the liver and then dumped, secreted into the bloodstream, and they stay there to execute important function, the albumin protein. Crystalline gene, crystal. This particular protein is important in our lens function in the eyes. It's a kind of lens protein. So no wonder that this crystalline protein is very important in the eye function, and only expressed, that protein is made in the lens, our lens tissue. So this albumin versus crystalline, two genes. But as you can easily guess correctly, these two genes are everywhere. It's not something that the albumin gene is only present in the liver. No. Like, let's compare. The albumin protein is only made in the liver, whereas this crystalline protein is only made in the lens tissue of the eyes. But that doesn't mean that these particular corresponding genes are only present in respective tissues, but they are everywhere. The difference is, respectively, they are only active in that particular tissue. So this albumin gene is only active in the liver, and this crystalline gene is, on the other hand, is silenced, inactive in the liver, and vice versa is true. This albumin gene, even if it is there in the lens cell, this albumin gene is not active, while this crystalline gene is active. How? RNA polymerases are there in both the liver and the lens tissues. The difference is availability of the transcription factors. So, in the liver cell, somehow, transcription factors are only available for this albumin gene. And as you can see, no transcription factors are around on the crystalline gene in the liver cells. And no wonder then why only albumin gene is expressed to make albumin protein only, while crystalline protein is not made at all. Completely opposite scenario can be observed in the lens cell, okay, of the eyes. So this albumin gene area, no transcription factors are available, whereas this crystalline gene area, lots of transcription factors are there. This kind of, maybe you can think about why not this transcription factor can also visit this area. Good question, because actually transcription factors are different. Some transcription factor can only work on particular gene, which means the difference between these two different type of tissues are availability of a particular transcription factors are the one that makes the difference. So, although it is very, very simplified version of the explanation, but still to some of you, even this one is not an easy concept to understand. So I will just simply stop here. I'm not going to go further about more detail. The mechanism will simply just work counterproductive, but simply this is the basic principle, and by which this differential gene expression can be explained. And you can also expand this in this the example into the version of this developmentally specific gene expression too. Timing, although these differences were presented in different tissues, but you can also imagine easily that the different availability of transcription factors can be manifested in terms of timing of your growth. Then you are now then seeing the example of developmentally controlled differential gene expression too. Okay. Now, okay, now it's the business of the mRNA intron thing. So, yeah, another cartoon explaining this RNA, so to say, processing. Basically, introns are all removed, and the remaining exons were put together. In general, we call such process as a RNA splicing. This RNA splicing, or more broader, taking into other features, or sometimes we call that as a overall eukaryotic messenger RNA processing, whatever. Okay, but why introns? Because introns doesn't seem to be needed anyway, but it's still in the middle of, inserted in many of, the vast majority of our all eukaryotic genes, whereas prokaryotes don't have such introns. So maybe these introns are some kind of advanced feature, because we are more evolutionarily advanced. The eukaryote and prokaryote, they are primitive, so you don't have them. So I do have them. So it must be, introns must be something good you guys don't have. Na poo poo. What? No. Actually, what happened is prokaryote, they are wisely dumped these introns. The evidence? Archaebacteria. Archaebacteria have introns. Wow. Prokaryotes don't have, the bacteria, sorry. So among prokaryotes, archaebacteria still have the introns like us, whereas bacteria, all the way, they somehow wisely dump all those introns, and then they have, now they are living much simpler, more straightforward way of lifestyle. But anyway, let's get back to the original question. Why do we still have this? There are two actual conflicting school of thought arguing about the actual origin of this intron, so that, we cannot really discuss their version of argument, which is it's a moot point. Regardless of their origin, still no one can explain why do you still keep this garbage, right? But at least this is something that we can have some kind of benefit by keeping this intron, is because this is something that obviously we have realized after the completion of this human genome project. When we finished reading our entire genome DNA sequence, before, scientists have some kind of estimation, because they already knew how many different proteins we have already. So roughly, say, it was 100,000 different proteins were there in each different human cell. Like whether each of 100,000 proteins are actually made or not is a different story, but at least potentially, when we counted them in every different places, like, it's up to at least 100,000 different proteins are there that were found. Then, so if 100,000 proteins, then the number of genes in our human genome must at least exceed the 100,000, right? Like, even if we do not have any other non-coding RNA, at least coding RNA should match the number of proteins. So we should have at least 100,000 genes. And if we put also, if we consider non-coding gene, it should be something like 200,000 genes. That's how the scientists have estimated that before the completion of our human genome project. Guess what? How many genes we have identified after completion? 20 freaking, oops, 23,000 genes? With this, are we Jesus Christ? With 23,000 genes, we make generate 100,000 genes? How? Doesn't add up. So that's where this so to speak, differential splicing. Hey, go away. Like the whole term is called officially called the alternative splicing. One real life example of such alternative splicing is there, but probably I should have actually provided more intuitive, straightforward diagram. But actually, this is what's really happening over here. This is one particular gene. All each different block that can potentially function as exons are labeled. Exon number 1, 2, 3, 4, 5, 6, 7, 8, 9. So all these numbered block areas are exons. But what happened? Depending on different location or different environment, some occasion you have exon 1, 2, 3, 4, 5, 6, 7, 8, 9, and then make a one protein. Some other times what happened? You have exon 1, 2, 3, 4, 5. Oh, oops, where is it? Number 6. Exon number 6 is missing. And then you produce different version of protein. And this is really widespread. Like, so these exons, not all exons are equally taken and then always included in the final version, but sometimes some exons are deliberately left out. Okay. Later on, see you later, and so you have more room for generating different versions of proteins out of single gene. In this way, a single gene can generate several different versions of a protein. That's how you can generate 100,000 protein out of 23 only genes. And that's what we call alternative splicing. And this is not only something that case we have found in human, but everybody, including plants, animals, fungi, and all eukaryote cells, these alternative splicing things turned out to be widespread. So at least still we don't know why we keep still this intron, but at least by keeping this intron, at least we can do this kind of juggling around things. In other words, sometimes intron can become exon. Some other times exon can become intron by simply just switching things. Central dogma, okay, it's a very good time, very good point to stop. So, yes, you get the early release today. So, have a nice weekend.